in Sanger Center's sequencing data. And for some of these that were known somatic mutations, you could always go looking at links to Cosmic, like this one here, and it would take you to Cosmic so that you can see whether the mutations were discovered in any of the other systems. So one of the advantages of having all these projects in place is we will be able to see actually how the other project, ovarian cancer project, look like for the ovarian P50 mutation. And here's an example that we can actually have the cosmic ovarian cancer, which is in a pack mode. This one summarizes the numbers of the samples that have some mutations and displaying them along with those that do not. So we actually are seeing that cosmic ovary, we have very few, 2 out of the 45 that have somatic mutations for PP53. This is a lot lower than what we are observing in TCG ovarian cancer. So these differences are indicating that the sample, giving people clues about whether the sample selections in the public database versus the current TCG project are different. And as ovarian cancer, we know that there's different ways of going through the disease process. Another way of looking at the use of next-gen sequencing data, like over here, this is a very strange mutation that's a silent mutation in displayed by next-gen. If we for the view of next-gen viewer, we are seeing this silent mutation probably is misannotated and really should be considered as a splice site variant. And again, we are seeing this one will appear to be homozygous changes across all samples in tumor, across all reads in tumor. So this is another way of looking at integrated information is looking at the integrate track for the Cancer Genome Workbench. So selecting the TCA ovarian integrated track, which is down here in full mode, allow a user to evaluate whether the homozygous mutant alleles in TCG ovarian sample are caused by deletion or caused by copy number deletion or caused by LH. And over here we are seeing, yes, there's some low level of deletion events happening in these samples marked with blue, but the majority of the samples actually were having close to copy number neutral events. And we can see some of these copy number neutral, like in this case, the copy number is 2.02, and we have a homozygous allele, and clearly there's a copy number neutral LH that leads to selection of the mutant allele in the tumor sample. And we always use another function of sorting out tumors to a specific order. So, for example, we select the top 4 samples and give it a name, just say test sample group. Then you will be able to actually group the samples consistently across all these data sets in order. That way, as you can see, that we allow us to compare all the data with the same order, and this is actually a very, very useful feature. Another way of looking at the integrate track is we could do a full pack mode that actually separates the exon array data with the rest of the copy number changes. So over here, the exon array data was labeled at the bottom, and the top one is the copy number data along with the mutation data. So in cases where there are somatic alterations that lead to intensive splicing, people actually can see different coloring of exons indicating differences. Again, all the detailed help for copy number for exon changes and for various information are over in this help button here. The second topic that might be interesting for an advanced user is to look into how to use the heat map views of this, and that will be covered in a different segment.